Good morning students. So today we have to start one more concept in the paper 2, that is biotechnology. Already we completed the genetics. Okay, so we have to move to the next concept in the paper 2, that is the biotechnology. So what do you know regarding the biotechnology? Biotechnology means what? How you will define here? So here biotech is nothing but it is the manipulation of living organism or parts of the living organism to make products useful to human beings. With the help of the living organism cells or organs, we have to manipulate that cells and organs to produce the useful product which will be useful for the human beings. Okay, means what? The cells or the tissues or the organs of the living organism that we have to manipulate to produce certain products, and those products will be useful or benefits for the human beings. So that we called as the biotech, or we can also call as the manipulation of the genes of organism to alter their behavior or characteristics or values. So the cells of the living organism that we have to manipulate, or we can alter the genes of that organism so that we can change the behavior or characters or value of that living organism. Okay, or we can also call as cell and gene technology used to produce new characteristics in plants and animals. Okay, so genes or the cells plays important role, or we can use the genes and cells to produce new characters in the plants and animals. So this everything is happened due to the presence of the biotech. Okay, biotechnology field. So genes, DNA, RNA, all these will be plays important role to produce the new characters or new products, or we can alter the behavior of the organism. So this everything is happen in the biotechnology. Okay, so next, timeline of the biotechnology means how the biotechnology field has been developed, how it is evolved. So startingly how it was, and today's biotech what it will be say. So in the 8000 BC, before that, the domestication of the plants and animals will be takes place like this in the first slide. Okay, so second 400 BC, before that, Egyptians master the art of wine making. So they know the art of wine making. So in the year that we called as the before Christ year or the era, Egyptians used to yeast to make the bread. Here they used yeast to make the bread. And in the 2000 BC, again Egyptians and the Sumerians learned brewing and cheese making. So how the year has been developed, like that the biotech field also developed. In the 500 BC, moldy soybean curds used to treat boils. Okay, this everything is the biotechnology, because here microorganism, yeast, fungi, bacteria plays important role to produce the products here. Here 300 BCE, Greeks develop grafting techniques. Okay, so how to make the grafting of the plants, so that techniques has been developed in the 300 BC era. So 1675, Anton van Leeuwenhoek discovers protozoa and bacteria. In the year 1675, the scientist known as Anton van Leeuwenhoek, he developed the microscope, and again he discovered the protozoa and bacteria. Then 1797, Edward Jenner created the cowpox vaccine. Then how the year gone, like that different work has been done due to the biotech field. So development has been taken place very fastly. Then in the year 1802, biology first appears. Okay, so in the second slide, proteins are discovered in the 1830. Okay, so firstly biology has been appears, proteins are discovered. Then in the 1859, Charles Darwin, they proposed the theory natural selection. Then in 1861, Louis Pasteur develops pasteurization. Okay, like this each and every process has been developed in the different years. So this we call as timeline, how in the different times, in the different eras or the different years, different discoveries has been done. In the 1865, Gregor Mendel and laws of inheritance. Okay, this is also we call as the genes or genetics. Then 1888, William Gottfried Walder discovered the chromosomes. Okay, so like this has been done. Bacteriophages were discovered in the year 1915. Then in the 1919, biotech was introduced by the Caroli Ericki. He was the scientist discovered the word biotech. Then in 1922, Frederick Charles Best discovered the insulin, which plays important role for the maintenance of the blood sugar level. Then Hermann Muller, radiation caused the defects in chromosome. Like that, various work has been done in the biotech. Okay, so 1953, that is Watson, Crick and Wickens described the 3D model of DNA in the year 1953. Then in the year 1966, the genetic code for DNA is cracked. Okay, how the genetic code plays important role, that has been discovered in the year 1966. Then 1971, the first complete synthesis of gene occurs in the year 1971. 1973, Stanley Cohen and Herbert Boyer perfected genetic engineering techniques. Then in the 1981, first transgenic animals are produced. Transgenic animals means transfer of the genes takes place in the mice, then that mice we called as transgenic mice. Okay, which character we want, that character gene we have to introduce, so that we called as transgenic animals. Okay, then polymer chain reaction technique has been developed in the year 1983. So like these, in the different times, different work has been done, that is genes, RNA, DNA, all these plays important role in the biotech. Okay, biology plus technology. Here technology plays an important role to produce the different products, and those will be helpful for the human beings. For example, insulin, that hormone is produced due to the biotech field. Growth hormone, okay, antibodies, vaccines, all these are produced in the field biotechnology. Okay, how they are produced? With the help of cells, organs, tissue, genes, DNA, RNA, proteins, all these that we call as manipulation. By the manipulation of these cells, genes, the products were going to be produced in the biotechnology, and those will be helpful in the future human beings. So types of the biotech. So which are the different types of the biotechnology? So first one is the microbial biotechnology, agricultural biotechnology, animal biotechnology, forensic, bioremediation, aquatic, and the medical biotechnology. Like these different fields or the types will becomes under the biotechnology. Okay, so first we shall see the microbial biotechnology. So the microbial word refers to the microorganism. Here we have to manipulate the microorganism like the yeast and bacteria. Those yeast and bacteria helpful for the production of the different bakery products. Okay, then firstly we have to manipulate the yeast and bacteria, then we can able to produce the bakery products here. Okay, so how these microorganism plays important role means to create better enzymes. Some means enzymes plays important role in the fermentation processes. Okay, so with the help of microorganism we can create the better enzymes that we called as, that will be possible in the microbial biotechnology. More efficient decontamination processes for industrial waste product removal. So contamination we can be removal of the contamination can be takes place with the help of microorganism in the industrial level. Okay, so contamination we can able to remove, that we can make the decontamination with the help of the microorganism. Used to clone and produce large amount of important proteins used in human medicine. We can able to also produce the proteins, those plays important role in the human beings. So this is regarding the microbial biotechnology. So second or the next is the agricultural biotechnology. Then in the agricultural, how the biotech plays important role here? We can able to produce the cloned plants, cloned animals. Okay, cloned plants means I told which character we want, that character gene we have to introduce, then that plant or animals we call as transgenic animal plants. Like that, cloned animals and plants also will be produced. By single cell or by the single organ or tissue, we can able to produce the entire organism here. Okay, so plants more environmentally friendly that yield more per acre, means with the help of genes or the genetics or biotech, we can produce the more yield in the agricultural field. Then second, resistance to disease and insects. So some pollutants will be cause the disease or that will be harmful to the yield or the crop, so that we can able to remove those pest or anything, so that we can able to produce the different what we called pesticides or the insecticides. Okay, so that we call that is possible in the biotechnology, that we call the resistance to disease and insect. With the help of biotech, we can able to produce the pesticides, insecticides. So again, we can able to produce the plants which can be produce more crops here. Okay, it will be gives the more yield. Such type of the plants we can able to produce in the biotech. Drug developed and grown as plant product. Drugs we can able to develop, and that is used as a plant product. These better plants ultimately reduce the production cost to help feed the growing world population. So population is growing on, so that we can able to produce more and more crop or more and more yield here. Then animal biotechnology. So animals are as a source of medically valuable proteins. Antibodies we have to produce, transgenic animals, these all we can able to produce in the animal biotechnology. Okay, antibodies means I told different antibodies which will be plays important role to fight against the antigens, to fight against the foreign particles. Okay, so that we can able to produce the antibodies, that is possible in the biotech field. Transgenic animals, that I told, transgenic mice, transgenic sheep, transgenic goat. Okay, different transgenic animals are produced in the animal biotech. With a single cell we can able to produce the whole organism, whole animal, that we called as the transgenic animals. Then animals as important models in basic research. Gene knockout experiment, different experiments has been done in the animal biotech. So animal cloning means that we called as the Xerox copy of the organism. Same organism we can able to produce, that is the cloning process, that is possible in the biotech field. So next is the forensic biotechnology, that is the DNA fingerprint. Okay, that victim or the anything is the means if the disease occurred, that can be able to identify with the help of DNA fingerprint, or the paternity cases, like these all different cases we can able to identify with the help of DNA fingerprint, and that is possible in the forensic biotechnology. DNA or a sample of the blood, hair, like this, if we got anything, so that we can able to identify the victim here. Okay, so next is the bioremediation. So bioremediation means what? The use of biotechnology to process and degrade variety of natural man-made substances. So many different man-made substances are there that should be degraded here. Okay, that is possible with the help of bioremediation technology. So stimulated growth of bacteria that degrade components in crude oil. Okay, the crude oil that can be degraded by the bacteria. So like this processes happens in the bioremediation. Adding nutrients to stimulate growth bacteria to clean up oil spill. So bacteria will play an important role to clean up all these oil spill. So just see the pictures how they cleaning here. Here for the cleaning purpose, bacteria is the important. That bacteria can be produced with the help of biotechnology. Aquatic biotechnology. So aquatic biotechnology means aqua, water. Okay, that we call as aquaculture. Raising, finishing or shellfish is controlled condition for use as food sources. These all plays important role for the protein purposes. Okay, so we can able to produce large number of fishes with the help of the biotech field here. So genetic engineer, disease resistant strain or oysters. So for that disease should not occur. Like those different fishes we can able to produce here. Medical biotechnology involved with the whole spectrum of human medicine. Preventive medicines, diagnosis of health and illness, treatment of human diseases. So in different cancers or any disease occurs, that can be identified with the help of different biotech fields here. New information from human genome project, gene therapy, stem cell technology. Okay, so any disease we can able to cure with the help of gene therapy. How the disease occurs due to the malfunctioning of the gene or malfunctioning of the protein. If the gene is not working properly or the protein is not working properly, immediately disorder or disease will occurs. So that gene we have to remove and we have to add the functional gene. That is possible with the help of gene therapy. Stem cell technology means embryonic stem cells or those stem cells we can able to culture, that we called as stem cell technology. So then which are the uses of biotechnology? Different uses are there, variety uses are there. First, in the field environment, development of medicines means antibodies, insulin, proteins. Okay, different products are produced that we call as medicines. Cloning will be possible. Same copy of the organism we can able to produce, that is the cloning. Gene therapy means non-functional gene we have to remove and we have to add the functional gene, that is possible in the gene therapy. Genetic testing, which disease occurred due to which gene, that we can able to do in the biotech field. Agricultural development. So these are the different uses of the biotechnology. Development of medicine. So microorganisms can be used to make biological substance for humans, which are equally effective as natural substances. Microorganism plays important role to produce most of the drugs, insulin, interferons. Okay, so treatment of diseases like hepatitis, arthritis are made possible with the help of biotechnology. So these all diseases we call as the genetic diseases. They occurred due to the defective genes, so that can be able to make cure with the help of the biotechnology or the genetic field. So different instruments are there, tools are there, then again techniques will becomes how to cure diseases. So many different work has been done in the field biotechnology, okay, which is the very vast field. Drug production, insulin, which maintains the blood sugar level. So this insulin production will be takes place in the biotech field. Human insulin is being produced using genetic engineering technique. So one technique that we called as genetic engineering technique. With the help of this technique, insulin is produced, human insulin, that we called as humulin, and it is used for the treatment of diabetes, that is low sugar level in the blood. Role of the insulin, that will be lowers the blood sugar. So if the person is unable to produce more quantities of the insulin, then that person should take the extra insulin from the outside. Okay, supplementary insulin that he have to take, he or she. That's why insulin production has been taken place in the biotechnology. Then interferon. So this protein is produced by the biotech field. Interferon produced using the recombinant DNA technology to treat the cancer patients. Okay, so this protein or this is so viral genome from one cell to another cell. So due to the abnormal cells or the means a continuous cell division takes place, those we called as the abnormal cells. So that we have to remove, or that removal has been taken place due to the interferons. So that is interferons produced by the biotech field here. Then that cancer patient can able to survive or cure, means bit cure will be takes place due to the interferons here. So next is the drug production, human growth hormone, okay, HGH growth hormone. So that is produced due to the biotech field. So treatment of dwarfism. So some people are the dwarf due to the low production of the growth hormone. So that treatment has been done due to the human growth hormone by giving extra hormones to that human being. Okay, if the person suffering from the dwarfism, then we have to give the growth hormone. So that growth hormone is produced in the field biotechnology. Gene therapy, that only I told, the non-functional gene we have to remove and the functional gene we have to add to cure most of the diseases. Okay, so that is the gene therapy. So here they given the process in which a faulty gene is removed or replaced with its healthy copy to restore the normal function of that gene. this is the gene therapy, how the process will be takes place. So that I will means gene therapy concept is there for you. So in detail that we have to study the next time. Okay. Then genetic testing, newborn screening, carrier testing, sickle cell anemia, these all are the genetic disorders. So how that diseases occurred, due to which gene, that testing has been done in the genetic testing techniques. Okay. So these all we called as the genetic testing procedures. Genetic testing can be used to detect the mutations regarding genetic disorders like cystic fibrosis. These all are the genetic diseases. Then how that disease occurred, due to which gene or the mutation has been taken place, what it is happened. So that entire thing has been means testing has been done in the genetic testing. Okay, plays important role in the environment or to remove the pollutants from the environment. So cloning, that I told, production of the exact copy of the organism, just like a Xerox copy, that is possible in the biotech field. Clones are organisms that are exact genetic copies. Every single bit of the DNA is identical. Each and every morphologically, genetically, that organism is similar as compared to the original. Okay, so agricultural development, increase the quality and nutritional value of the plants. So this entire thing is possible in the biotechnology. Okay, up to this regarding the introduction of the biotechnology and types of the biotechnology. So next class we have to study the next concept in the biotech. Okay, thank you.